Okay, so a couple quick explanations. This is the kind of the second part of our gel electrophoresis conversation. So I'm going to use this image here. Just pulling this up on the computer. So okay, so here's how this works. So we talked a little bit about the box and how the box works. So there are going to be these small holes or wells. When the DNA goes in these wells, let's say we have sample 1, 2, 3, and 4 from left to right, what's going to happen is the samples are going to move through the gel when the electricity starts. In this particular one here, you're seeing that the fragments have moved, 1, 2, 3, 4, 5 fragments. So there were 5 cuts on this particular piece of DNA, 2 cuts, 1 cut. So there were, if we talk about the restriction enzymes, if there were 5 cuts, I'm sorry, 5 pieces. There were 4 cuts with the restriction enzyme. So one piece of DNA that was cut 4 times to give you 5 pieces. In this case, there were no cuts, so there was no match or no AGCT match with the restriction enzyme that was used. Here, there would have been 1 cut, 1 cut. Okay, the size of the lines represent the size of the fragments. So, for example, the further away the lines or these bars show up, the smaller the fragments. So this fragment here would have been the smallest. The ones way down here would have been the biggest fragments. All right. Once again, largest to smallest. Why? The fragments get stuck in inside the gel. I want to show you this other picture here. So this is this is an actual gel. So what they did with this particular gel is there's a couple of things going on. Here are the wells right here. They use a glow-in-the-dark stain that glows under UV light. This is usually how they do this. And what they're able to do is on either side, so on this side and on this side, notice the lines are exactly the same. And the reason why is they created what's called a standard. So in the standard is they know the size of the fragments that they're starting with. So the fragments that range in size from 100 to 2,000. Notice the 2,000 means number of bases. Notice the longest ones get stuck the furthest. So when they have unknown fragment sizes, they're able to use a standard, or think of the standard as like a ruler for this. So in this particular sample that I put here, it had all of these sizes here. And these are known sizes because they're known pieces of DNA. All right. Excuse me. One last image I want to show you. It just happened all these images are actually kind of cool in the sense. So once again, here's our power supply. Here's our gel. We put some type of buffer on top, put our samples inside, run the electricity, and it pushes the DNA through the gel. So I want to show you this particular question. This was in 2007. The most common. So here's what they've got here. We're going to look at a couple different things. What you're seeing here is a copy of the gel electrophoresis results. If you remember the first picture we saw here, if we go back to this picture here, right, the standards, this here on the side, these are the standards. So these are the known sizes. So they took in the first well, which would have been down here somewhere, they would have put the standards 1 through 10 in 10 increments. So here's a 100 piece, a 90 piece, an 80 piece, a 70 piece, so on and so forth. Then they took and they used 2 different restriction enzymes. The restriction enzymes are named for the bacteria that they come out of. So EcoR1 and HAE3 came from bacteria. So this particular bacteria, EcoR1 or E. coli restriction enzyme 1, cut a piece of DNA into 2 pieces. One section was 70 long, one was 30 long, for a total of 100 base pairs. You took the same DNA and cut it with HAE3, you get a piece that was 60 and 40. When you use both enzymes, you get a 10, a 20, a 30, and a 40 piece. What they're looking for you to do is create, and they're talking about, when they talk about this, they talk about bacterial plasmids, which is 100 kilobases in length. Sorry, I'm in the basement. Somebody just got in the shower, so you might hear a little bit of water running through the pipes. Anyway, so the bacterial plasmids here are 100 kilobases in length. If you look down here, this diagram, this template, is supposed to represent the circular DNA found in bacteria, 100 kilobases in length. What they're looking for you to do is to draw a map of where the relationship between these pieces, and we'll talk a little bit more about this next time we meet online. See what you can do with this. It's interesting. It's got some This is a very difficult question, number 4. So give this a try, and I'll touch base with you guys tomorrow. All right? Have a good day.